So today what we are going to be working on is your first set of selective and differential media. And so I am going to be inoculating, there's a total of 8 different types of selective and differential media. I will be doing 4 today, and then we'll be doing the other 4 next week along with reading the results. So the first test that we are going to start out with is the gelatin test. And like I said, all of this is in your lab manual, so if you want to follow along or just, take notes from me talking, you can definitely do that. So the first test that we are looking at is the gelatin test, or GEL for short. And so, well, before we go over that, let's go over what selective and differential media actually is. So selective and differential media is media that we use to try and identify an unknown bacteria. So the rest of the semester, that's what we're going to be working on, doing tests to try and identify an unknown. So we've already went over all of the stains that you would be doing. Now we're going to go over the different types of biochemical tests that you would be using. Selective and differential media, first we need to go over what selective is, then we need to go over what differential is, then we'll do a combination. So selective media is exactly what it sounds like. It is media that is only allowing certain things to grow. It has what is called a selective agent in it that is only allowing certain bacteria to grow and preventing growth of unwanted bacteria. So, we don't have any, we have an example of selective and differential media today, but next week will be the selective media. Now, differential media is exactly what it sounds like. It's showing differences in the bacteria based on what kind of metabolism the bacteria is doing. Okay, so it has in it what's called an indicator. So based on the metabolism and based on the products that are produced from that metabolism, that indicator is going to change colors. And then based on the colors that you see, that's going to differentiate between a set number of bacteria. And we have multiple examples of differential media today that we'll go over. And then you can have a combination of the two, selective and differential, which we have an example of that today, where it has a selective agent and an indicator in it. So we're going to get, not only is it going to select for certain bacteria, but then between that bacteria, it's going to differentiate. So it's going to give us lots of information about our unknown. So now that we've went over that, now we're going to start going over each test. So the first test, like I said earlier, is the gelatin test. This is a strictly differential type media. Okay, so there is no selective agent in it. Pretty much anything is going to be able to grow on this plate. The substrate in the plate is called gelatin. That's where the name of the media comes from. Okay, so gelatin is the substrate, and we are asking, does the bacteria have the ability to eat the gelatin, yes or no? Okay, basically, does it have the ability to break the gelatin down? If it has the ability to break the gelatin down, it produces an enzyme called gelatinase. So if the bacteria can produce gelatinase, that will break the gelatin down, and then the bacteria will take that in and use that as a nutrient source. Okay, so basically it can eat the gelatin. Some bacteria can do this, some can't. So we are going to inoculate gelatin media, and then I'm going to incubate it for 24 hours, and then we're going to see if the bacteria can eat the gelatin, yes or no. You'll notice that there's an indicator that needs to be added to the plates, and that indicator is hydrochloric acid. Now, my gelatin plates, the way that they're made, we don't actually have to add the HCl to the plates. That is kind of an what we used to do a while back. So I'm not actually going to have to add the HCl to these plates to be able to see the halo, but you still have to know that that is a potential indicator that needs to be added sometimes. So a positive result is a halo of clearing around the bacteria. A negative result is no halo. So basically, if the bacteria ate the gelatin, you're going to see an area around the colonies where there is no gelatin left. It ate it all. So you're going to see a halo of clearing. If the bacteria cannot eat the gelatin, then all of the gelatin is still going to be on the plate. You're not going to see a halo of clearing. Okay? And that's the way that gelatin works. And here's an example of a gelatin plate. Okay? And I'm going to inoculate these for you, and I'll show you exactly how to do that. And they're very white, and that white color that you're seeing is the gelatin on the inside of the plate. Okay, so That's why they're that very white color. All right, so the next media that we are going to look at is called starch, okay? And this, again, just like the gelatin, is a strictly differential media, and it's looking to see, can the bacteria break down or hydrolyze starch? Okay, can it eat starch, yes or no? Now, starch is a polysaccharide. It's a sugar. It's a very large sugar, okay? So the bacteria has to be able to break the starch down into the glucose monomers before it can bring it into the cell and use it for any type of metabolism. So to do that, the bacteria has to produce an enzyme called amylase. Amylase is an exoenzyme. So the enzyme is made on the inside of the cell, then sent out of the cell to break the starch down. Okay, so that's what I have here. Name of the media, starch, STA. Starch is our substrate. That's what our bacteria is potentially breaking down. Okay, and then it has to produce the enzyme amylase to be able to do that. So just like with the gelatin plate, our question is, can the bacteria eat the starch, yes or no? Some can, some can't. So the indicator that has to be added to these tests is iodine. So iodine reacts with starch and turns black. So if the bacteria ate the starch, there's not going to be any starch around the colonies. And you're going to, again, get a halo of clearing. If the bacteria did not eat the starch, all the starch is going to be there, and you are going to see a solid black plate. So the reason why I like to go over gelatin and starch back to back is because these are the ones that they're very similar. Okay, they're both strictly differential. They're both having to add an indicator. They both have a halo for a positive result, and they're asking the same questions. Can the bacteria hydrolyze the gelatin or the starch? Okay, so whenever I remember these tests, I always group gelatin and starch together because they're very similar. Okay, so if that would help you while you're studying for your quiz or for your final, that's a way to help remember it. Now, the next test is called MSA, mannitol salt agar. Okay, that's what the MSA stands for, mannitol salt agar. This is a both selective and differential media. So of these four, it's going to be the most complicated because it's both selective and differential. The substrate that's in the media is mannitol. Mannitol is a sugar alcohol. It's a simple sugar, so it's small. The selective agent that's in the media is salt, 7% salt. And this high percentage of salt is selecting for staph. Okay, so you would use this media if you potentially thought that you might have a patient that has a staph infection. Okay, so if you did a gram stain and it came out as a gram-positive cocci that formed clusters, then you would say, okay, I think that this might be a staph infection. You would then take a culture of it and streak it on MSA. If it grows on MSA, then that means that it is some form of staph. Okay, that's the selective part. It's selecting for staph. Other bacteria cannot live in a high salt concentration. That's why your mother told you to gargle with salt water, because the salt kills the majority of bacteria. Now, the indicator that's in the media is called phenol red. This is the differential part of the media. So, say we take a culture, streak it on MSA, and it grows. That tells us that it's some form of staph. Now, we need to figure out if it's a pathogen or not. Pathogen being Staph aureus, non-pathogen being Staph epidermis. Okay, Staph epidermis is on everyone's skin. It's not harming us. For the most part, it's not pathogenic. And so that would be the non-pathogenic strain. Staph aureus, of course, would be the pathogenic strain that could cause problems. So we want to see, is this a Staph aureus or is it a Staph epidermis? To do that, we need to see if the bacteria can ferment mannitol. Okay? So let's talk about fermentation versus respiration. For fermentation, the bacteria takes the mannitol and turns it into acids. Okay? Acidic fermentation. So it takes the mannitol, turns it into acid to produce a little bit of ATP. Not all bacteria can ferment the mannitol. Respiration is where the bacteria takes the mannitol, does glycolysis, Krebs cycle, puts those electrons through the electron transport chain, and ultimately makes water. So you have fermentation, which is acidic end products. Then you have respiration, which is basic or neutral end products, water. Okay? So there's a huge difference there. If acids are produced, your pH is going to go down. If water or basic end products are produced, your pH is going to go up or stay the same. So that's what we are looking at. Staph aureus has the ability to ferment the mannitol, whereas Staph epidermis does not have the ability to ferment. It uses it for respiration. So to see which one we have the pH indicator. The pH indicator being phenol red. Under acidic conditions, the phenol red turns yellow. It's already red. Here's my MSA plate. That's what it starts out as. So if I streak the bacteria, it grows, and this plate turns yellow, then that tells me that the bacteria that I have is Staph aureus. It fermented the mannitol, and it was able to grow. If the bacteria I streak on this plate grows, but the plate stays red, that means the bacteria use the mannitol for respiration, producing water and basic end products, and the phenol red stays red under basic conditions. And you're going to get to see examples of both of those when we look at the results. So not only for the MSA, you have to look at the selective part of it. Did it grow, yes or no? If it grew, then you have to look and see if it fermented the mannitol or not. Fermentation giving you a yellow result, no fermentation giving you a red result. Now, if you streak the bacteria on the MSA and it doesn't grow at all, then that's an all-out negative test. That means that this bacteria that you're dealing with is not a type of staph. It's going to be something else. And then you have to start all over again. So that's your MSA, selective and differential media. So it's a two-part. All right, now this last media is called SBA. SBA stands for sheep's blood agar. Sheep's blood agar. And this is a strictly differential test. So just like the gelatin and the starch, the blood is also a strictly differential test. So we don't have a selective agent, okay? And you also don't have to add an indicator to this, and you're going to see why in just a minute. This substrate is blood. So we are asking the question, does the bacteria have the ability to eat blood cells? Okay? Now, the reason why we do SBA is because of strep. Okay? If you've ever had strep before, a strep infection, you know that your throat hurts really, really bad. You have these massive white pus pockets in the back of your throat. Okay? That's because the strep is eating your blood cells. Okay? So that's why you have the absence of that red color in your throat and you have those pus pockets, okay? Because it's eating all of your blood cells. So that's one of the reasons why we do a blood agar plate. So let's talk about what the results are going to look like, because this is what's different about this media. There is no selective agent. So here's our sheep's blood agar, and it's of course red because it has blood in it, okay? Sheep's blood. So this is basically just TSA, which is a general purpose media, and then blood has been added to it. So if you streak, so you would take a throat swab. That's what you would do with this plate. You take a throat swab, streak it on SBA, and the bacteria is not strep. Okay, so it doesn't have the ability to eat the blood. But you get a greenish color. Okay, the colonies and the agar around them will be a green tint. That is an example of alpha hemolysis or incomplete lysis. Lysis meaning to break. So incomplete lysis or alpha hemolysis, these bacteria have the ability to suck the nutrients out of the blood cells, but they do not have the ability to completely consume the blood cells. And that's why it's called incomplete lysis, because they can still utilize or suck the nutrients out of the blood, still utilize the nutrients, but they just can't completely consume the blood cells. So this is called alpha hemolysis. And again, the result of that is going to be a green color. Now, if you streak the plate and it is a form of strep, it will do what is called beta hemolysis. Beta hemolysis is complete lysis. So the bacteria completely consumes and eats the blood cell. This is where you are going to have an absence of blood. Around the colonies, you're going to see a clearing, okay, where there's no more blood left because the bacteria ate all of it. If you see the clearing around the colonies, that is complete lysis. That bacteria can completely consume the blood. And the third option is gamma hemolysis. Now, gamma hemolysis is rare, and this is where the bacteria can't utilize the blood at all. So it does nothing. The bacteria will simply grow on top of the SBA. You will not see a green color, and you will not see a clearing. Like I said, this is very rare. I am going to inoculate our 4 different kinds of media. I will let them incubate for 24 hours, and tomorrow I will come in and make another video on what the results look like. Okay. You would need 8 plates total, 2 of each. So I have 2 starch plates, 2 gelatin plates, 2 MSA plates, and 2 SBA plates. Okay, let's see. Gelatin's first, so we'll change that order. Now, I've already have them all labeled. Of course, whenever you're inoculating media, that's the first thing that you want to do is you want to label all of it. Okay, so I labeled what's what and then what I'm putting on each of the bacteria. Now, the first set that I'm going to work on is the gelatin. Okay, now, if you look in your lab manual under the procedure, you're going to see that the 2 bacteria that we're using are Bacillus and E. coli. One's positive for gelatin and one's negative for gelatin. So we have a positive control and a negative control. Now, if you were doing an unknown, then you would, of course, do one for your unknown also. But you always want to have your controls, and that's what we're going to use today. Now, for the gelatin plates, remember I told you that you have to look for that halo of clearing, okay? So these are not going to be normal streak plates because I want to be able to see that halo. All right, so my gas is on. I'm going to use my loop for this. Anytime you're doing anything with plates, you're going to want to use your loop. So I'm going to flame it. Let it cool. Now, I'm using broth cultures for this, but you could definitely use liquid cultures. Solid off of slants if you wanted to. Normally, when you would be in class, you would be using the slants, but I had broth cultures, so that works just fine. So the first one I'm going to do is my E. coli, so I just simply dip into my bacteria, open my plate, and I'm going to just do a line straight down the center. Flame my loop again. Let it cool. I'm going to add this to a plate box for an incubate. Okay, the other one that I'm going to be doing is my Bacillus. So I'm going to mix this up a little bit. Do my line straight down the center. Now those are my gelatin plates. I'm going to go ahead and do my starch plates next. Again, these are inoculated the same way. It's still a line straight down the center. And the bacteria are also the same. I've got my Bacillus and I've got my E. coli. So I'm going to do a line of the E. coli and a line of the bacillus. So here's my E. coli. Let my loop cool some, and my bacillus. Alright, I'm done with my gelatin and my starch. Now I'm going to move on to my MSA. Now, the MSA and the SBA are both going to be regular streak plates. So, no, just line straight down the center. We're going to do regular streak plates. But we are going to need to do a nose and a throat swab for the MSA and then for the SBA. So, the MSA, I have 2 plates again. One is a nose swab, and one is Staph aureus. Okay, that's my positive control. And then my nose swab. Okay, so I'm going to go ahead and do my Staph aureus first. So this is a normal streak plate. So your normal streak plate technique, the T, the 3 quadrants, flaming my loop in between each quadrant. Okay, so here's my first quadrant. Flame my loop. Second quadrant. And I'm flipping my loop, and I'm going to do the third quadrant. Of course, you normally do not want to stab the agar, but sometimes it happens, so no big deal. Okay, now I'm going to do my nose swab. So, of course, we need a sterile cotton swab for this because I'm going to swab my nose. Then I'm going to use the swab for the first quadrant, get rid of the swab, and then finish the streak plate with my loop. Okay. Streak all in quadrant 1. This is going to go in biohazard. And then I'm going to finish my streak plate with my loop. Okay, so add that to my collection. And the last set are the SBA. And for this, I have to do a throat swab, and then I have to do Staph. Okay, Staph is my positive control. So Staph does the halo of clearing. Hopefully my throat swab will give me that green color, which is the beta hemolysis. Okay, so I'm going to do the Staph first. Go ahead and get that one out of the way. This is, again, the normal streak plate. Okay. And now for the throat swab. Now, whenever you do the throat swab, you want to we would normally want to make sure that you get all the way into the back of the throat, not hitting anything but the back of your throat. And now we're going to finish our streak plate. And that's it. So I've added them all to my box. Okay, there's all 8 of them, 2 times 4. Okay, and so I'm going to go put these in the incubator for 24 hours, and then tomorrow with the results. Okay, so we'll do the results, and then we'll inoculate our second half of our selective and differential media. So I am back with the results for your first selective and differential media. So we're going to start out by reading the results of the gelatin plates. Remember, that was the very first one on the list of the media. And remember, it is a strictly differential media, and we are looking to see, can the bacteria eat the gelatin, yes or no? So if you remember, I inoculated these plates with, these are my gelatin plates. I inoculated them with a line straight down the center of the bacteria. Okay, so it's kind of hard to see on here, but you can see that there is a line of the bacteria. And so we've got a positive and a negative. So remember, a positive result is the halo of clearing, where the bacteria has eaten the gelatin. And then a negative result is where there's no halo, so all the gelatin is still there. So let's look at a negative result. E. coli is negative for gelatinase. So as you can see, all of the gelatin is still there. Okay, there is no clearing or area where there's clearing. Okay. Look at the back of the plate, it's maybe a little bit easier. Okay, see, everything's white, no halo at all, so that's a negative result for gelatin. Now, a positive result for gelatin is bacillus. Okay, so I don't know if you can see that, but you see that area around the colonies where you're seeing that clearing. Okay, so there's no gelatin there. So a lot of times what I like to do is take my flashlight on my phone and actually kind of hold it. Okay, so I don't know which side's better. Actually, let's do this. So it makes it a little easier for you to see that halo. So you see the colonies going down the center. That's the white colonies. Okay, and then you see that area around the colonies where there's no gelatin left. That means that this bacillus produced gelatinase, went out of the cell, broke down the gelatin, and then it brought it into the cell. Okay, so it ate the gelatin. So that's that clearing. So here's clearing. That's a positive result. And here's no clearing, okay, which is a negative result. So that's my gelatin. Very straightforward, very easy. You get a halo, positive, no halo, negative. Now, moving on to the starch plates. So here's my starch plates. So here's my bacillus, here's my E. coli. Now these had a little bit of condensation on them. So normally you would want just a line straight down the center, but since they had some condensation on them, you got some growth around the edges where it was wet at. Same thing here. That's what that all is the condensation that was there. But that's okay. Okay, so remember, we have to add an indicator to these plates. Remember, the indicator for gelatin was HCl, but I told you we probably weren't going to have to add that. Like I said, we didn't add it. We were able to see the results. But the starch plates look very similar, right? It's like a clear color. So I've got growth of my bacillus and my E. coli, but I can't tell the difference. I need to add my indicator. My indicator is iodine. Remember, iodine reacts with starch and turns black. So for a negative test, if all the starch is still there, you're going to get a solid black plate. That's a negative test for starch. If you get an area of clearing where there's no starch there, that's a positive result for amylase, a positive starch for starch hydrolysis, meaning that there's no starch left. So I'm going to add my iodine to my plates. And I'm just covering the entire plate. And if we were in lab, you would be doing this with your plates, but we're not, so. All right, I covered these with iodine, and I'm going to let them sit for a few minutes. That way we can let the iodine react, and that way we can hopefully get our halo. So while those are sitting, I'm going to move on to the SBA plates. That's our other type of. Here's our SBA plates. Remember, SBA stands for sheep's blood agar. Okay? Our substrate here is blood, and we are looking to see, can the bacteria eat the blood, yes or no? So, remember, there is no indicator here. Okay? You either get a halo of clearing, that's the bacteria ate the blood, that's beta hemolysis. You get a green color, that's incomplete lysis or alpha hemolysis. This is where the bacteria just sucked the nutrients out of the cell but left the cell there. And then the third option is gamma hemolysis, and that's where the bacteria doesn't utilize the blood at all. It just grows on the media and doesn't utilize the blood. That one is rare. So we don't have an example of that because it is so rare, but we do have an example of alpha and beta. Okay, so I'm going to use my flashlight on my phone again to try to make this a little bit easier for you to see. But here's my throat swab that I did yesterday. Okay, and as you can see, when you shine the lights, there is a greenish color to the plate. That greenish color means that this is alpha hemolysis. So the bacteria did not completely eat the blood. It just sucked the nutrients out and it did incomplete lysis. So that's that green color that you see. So no strep throat infection for me. Because if I did have strep, when I did this, you would see large areas of clearing where the bacteria completely ate the blood. So there's my alpha hemolysis. So beta hemolysis is complete lysis, where you see clearing around the colonies. So if you look here for my staff, let's see if I can try and get this as good as possible. You will see in that large area, there is clearing. So my light is going straight through. Okay? You see that, where my light's coming straight through? That's that area of clearing that you're able to see. Against the black background. Okay? So, this large area, let's see, right here is what I'm talking about. So, you see it's clear. You can see my flashlight through it. It's completely clear. So staph is positive for beta hemolysis, complete lysis. So the staph is completely consuming the blood cells. Let's see if maybe it'll be easier. Okay, so I'm getting complete clearing. So this is beta hemolysis, complete clearing. And then this is alpha hemolysis, where you're getting that green color. So alpha, and then beta with the complete clearing. So now my iodine should be ready. So I'm going to bring these over to the sink. So I'm going to let these sit and just kind of let that excess iodine get off of there while we read our MSA plates, and then we'll come back to those. So our last set of plates that we have to read, other than the starch plates, are the MSA plate. So remember, MSA stands for mannitol salt agar, and these are the only ones out of these four that we did that are both selective and differential. Okay, so what that means is only certain things are going to grow, and then those things that grow, we're going to be able to differentiate what kind of metabolism is going on. So remember, the substrate is mannitol, and we're looking in the selective agent is salt, a very high concentration of salt. So if the bacteria grew, that means it is some type of staph, because only the staph can grow in that very high salt concentration. So if you look on both of my plates, I have growth. So here's my nose swab. As you can see, there is growth there. I've got colonies growing on that plate. And then here is my Staph aureus. Okay, as you can see that there's clearly growth on that plate. So that means that since I've got growth on both of them, both of these bacteria that are growing are some form of staph. That's the selective part. Now, the differential part is the bacteria a fermenter, yes or no? That's going to differentiate between pathogen and non-pathogen. Staph aureus, being the pathogen, can ferment the mannitol. When it ferments the mannitol, acids are produced, the phenol red turns yellow. So yellow and growth means that this bacteria has to be staph. If I get growth but I get a red color, that's staph epidermis. Staph epidermis is a non-pathogen. It cannot ferment the mannitol. So let's look at our results. Here's my nose swab. So like I said earlier, I've got growth on there. That means that it is some form of staph, but luckily my plate is red. So that means that the bacteria that's growing in my nose is staph epidermis. That's pretty much what everybody usually gets whenever they do the nose swab, is this, a red plate with growth. Now, if you were to swab your nose and get no growth at all, that just probably means you didn't do a very good job of swabbing, because pretty much everybody has staph epidermis somewhere on their skin or in their nose. Now, some people are carriers of staph. So if you've ever had a staph infection before, or you're simply just a carrier, sometimes we do actually get yellow plates from the nose swabs. Unfortunately, we didn't get to do that. That's usually a fun exercise. Everybody kind of gets to see what's growing in their nose, what's living in their nose and throat, but we didn't get to do that. So here's my nose swab. So I got growth, and it's a red plate, so that means this is staph epidermis. Now here's my staph plate. I got growth, so it's some type of staph. And of course, it's Staph aureus, so I get that yellow color. Now, the whole plate did not turn yellow just because the colonies did not grow everywhere. And that's just because I could have left it in the incubator a little longer, probably another 12 hours or so, and you would have gotten even more growth. But I wanted to go ahead and make the video for y'all. So that's why only half of the plate turned yellow. But where you've got that heavy growth at, the plate has turned yellow. So it grew, some form of staph. It turned yellow, so therefore it is Staph aureus. Now, if I streaked, say, E. coli on the MSA plate, it's not going to grow. Nothing's going to grow because it can't grow in the high salt content. So that would be a straight negative for everything. Not any kind of staph, and therefore not a Staph aureus or Staph epidermis. So that's the results for our MSA. Now, let's look at our starch plates. Remember, halo is positive for amylase. That means starch hydrolysis did happen. Negative is no halo at jet black plate. So here's our E. coli. E. coli is negative for amylase. So you see my plate is jet black. All of the starch is still there. Okay, so negative for starch hydrolysis. None of that starch is gone, so E. coli does not produce amylase. Here's my bacillus. Whoa, look at that. Do you see the clearing? Okay, I'm going to use my phone again. My flashlight. Look at that clearing. Okay. That means that this is bacillus, and bacillus is positive for amylase. It ate the starch. Now, like I said, these have not been in the incubator, but just barely 24 hours. So if I were to leave this out even longer, the bacillus would have more time and would eventually eat pretty much all of the starch that's in the plate. But you're seeing that halo around the colonies, and that's what we are looking for. So halo, positive for starch hydrolysis, positive for amylase. No halo. You can't even see my flashlight through that. Jet black, negative for starch hydrolysis, and negative for amylase. Okay, everybody, so that's it for all of our results for this first set of selective and differential media.